There are two stages of meiosis. There's meiosis 1 and meiosis 2. Meiosis 1 starts with prophase 1. At this stage, the cell is diploid because there are both sets of chromosomes within the cell, making the count 46. During prophase 1, the chromosomes condense and they line up with their homologous pairs. Like this. Okay. This is called a tetrad. Also during prophase, the crossing over occurs. Crossing over is the transfer and exchange of information between the homologous pairs. So like this would go over here. This would go over here. And like this would give this one. And this would give this one. Okay. Because of this, you will no longer have the exact same chromosomes. Crossing over is the reason for genetic variance. However, there are some genes that are linked, say like these two, and they usually don't get separated. Crossing over is the only time that these linked genes can be separated if they get cut in the middle of them. So say when they're crossing over, these two linked genes get cut in the middle. And so when they're crossing over, and this gives it 1, now these linked genes are no longer linked. And then while all of that is happening, the centrioles form and they start to move to the opposite sides of the cell.
In metaphase 1, the tetrads line up behind each other in the middle of the cell. Then in anaphase 1, the homologous chromosomes move apart. Finally, in telophase 1, the cytoplasm divides during cytokinesis and the chromosomes decondense. Now, there are 2 diploid cells. After telophase 1, meiosis 2 starts. The first step in meiosis 2 is prophase 2. In prophase 2, the chromatin recondenses and there are 23 pairs in each cell. Also in this stage, the centrioles move to the opposite poles. In metaphase 2, the pairs in each cell that line up behind each other. In anaphase 2, the sister chromatids in each pair are pulled apart to the centromeres. Finally, in telophase 2, each cell cytoplasm divides and therefore makes 4 haploid cells.
One, two, three, 4.
The end.